Approximately 97% of the water covering the Earth is ocean. Most organisms do not have the physical mechanisms to process and remove excess salt in their systems, resulting in dehydration and plasmolysis of the blood cells. So how do marine animals live in highly concentrated salt environments, and how do different species process excess salt in comparison to one another?
That's a little salty.
Over time, marine organisms have adapted cranial exocrine glands to serve as salt secretion glands that secrete a highly concentrated salt solution. These include the lateral nasal, lacrimal, lingual, and sublingual glands. Found in elasmobranch, seabirds, and some reptiles, salt glands are lobed containing many secretory tubules lined with single-layer epithelial cells. Active transport moves excess salt from the blood through sodium-potassium pumps into the salt gland. This process enables marine animals to digest seawater without having to eliminate excess salt through the kidneys. Location, diameter, and length of the salt glands is dependent on the species, diet, and their environment.
Lateral nasal glands are most commonly found in lizards and birds. These glands are located in or on the skull in areas of the eyes, nostrils, or mouth. The Galapagos marine iguana has salt glands positioned in the nostrils, which expel salt from the system via mucus. Amblyrhynchus cristatus is endemic to the Galapagos Islands and is the only extant marine iguana out of 416 species. This species is exposed to high concentrations of salt through its diet, primarily composed of soft-body macrophytic algae and its environment. The Galapagos marine iguana is incapable of creating highly concentrated liquid urine, limiting its urinary electrolyte excretion. As a result, this iguana must excrete excess salt through the lateral nasal gland by snorting and sneezing. This expulsion also acts as a defense mechanism to ward off predators and competition.
In seabirds, the lateral nasal gland is located in the nose. Seabirds sneeze and shake their head to expel excess salt, as their kidneys cannot properly process high concentrations of salt. The exclusive function of the lateral nasal gland is osmoregulation, the stabilizer of water and salt concentrations in the body. Unlike the lateral gland marine iguanas and seabirds, the lacrimal gland secretes an aqueous layer of tear film, responsible for all salt excretion and testudines. Located within the orbital cavity, the lacrimal gland secretes high concentrations of salt as tears, as the kidney in sea turtles is unable to process excess salt. The diet of marine turtles consists of a variety of marine invertebrates and plants, both of which have high salt concentrations. Both play an integral role in their adaptation to the marine environment.
In comparison, sea snakes have lingual glands present on the ventrolateral surface of the tongue sheath. This gland enables sea snakes to avoid excess salt accumulation by extending the tongue. Multiple ducts in the tongue sheath expel salt into the surrounding environment. Similarly, crocodiles have lingual salt glands located on the posterior half of the lingual surface, under the tongue. These salt glands appear as 20 to 40 distinct pores which secrete high salt solutions onto the tongue. In addition to having lingual salt glands, crocodiles also have renal salt glands to assist with salt secretion.
The evolution of cranial exocrine glands to serve as salt secretory glands play a major role in osmoregulation and homeostasis in marine organisms. The high salt diet environment of these organisms will result in dehydration and plasmolysis of blood cells without the adaptation of salt glands. Since the kidneys of these marine animals are incapable of processing excess levels of salt, these glands are vital for the survival of these animals.